Okay, so the microbes in human cell, right? Now you can see uh, so microbes are present everywhere in soil, water, air, inside our bodies, and that of other animals and plants. They are present even at sites where no other life form could possibly exist. Sites such as deep inside the geysers, thermal vents where the temperature may be high as 100 degrees Celsius, deep in the soil under the layers of snow several meters thick, and in highly acidic environments. Right. The microbes are diverse protozoa. So what are the microbes? So these are the diverse protozoa. So these are the diverse protozoa. What are the protozoa? Protozoa includes in the phylum in Protista, in kingdom Protista, in division Protista, right? So not phylum. So microbes are diverse protozoa, then bacteria, then fungi, then microscopic plant and animal, then viruses, viroids, and also prions that are proteinaceous infectious agents. So some of them, so all these are microbes. Okay, microbes are diverse protozoa, bacteria, fungi, macroscopic animal, plant, viruses, viroids, and also prions that are proteinaceous infectious, right? So microbes like bacteria and many fungi can be grown nutritive medium to from colonies from the colonies that can be seen with the naked eyes. So this can be seen with the naked eyes. Okay, such cultures are useful in studies on microorganisms. As you know, the bacteria in the first diagram, they rod shaped magnified, then second is the spherical shaped magnified, and rod shaped bacterium showing flagella magnified. You can see the rod shaped bacterium that has the flagella, and the second one is the diagram is the viruses, a bacteriophage. You can see head, collar, then tail, then plate, then pins, then prongs, right? So head collar, then tail, then plate, then pins, then prongs. And then in second diagram it is a adenovirus which cause respiratory infections. And the third diagram is the rod shaped tobacco mosaic virus, that is TMV, that is compact rod shaped viruses. So in the first diagram, colonies of bacteria growing in petri dish, and the second one is the fungal colony, it is fungal colony, and first one is the bacterial colony. So they causes diseases in animals and plants. Not all microbes are harmful. Okay, several microbes are useful to human in diverse ways. Some of the most important contribution now. So microbes in household products. So we use microbes or products derived from them every day. Okay, a production of curd from the milk. Microorganisms such as lactobacillus, other commonly called as lab, they grow in milk and convert it into curd. Okay, so during growth, lab produce acids that coagulate and partially digest the milk proteins, and small amount of curd added to the fresh milk as in column or starter contain millions of that is lactic acid bacteria, which at suitable temperatures multiply, thus converting milk to curd, which also improves its nutritional quality by increasing b12 vitamin b12 in our stomach too. In our stomach too, the lab play very beneficial role in checking disease causing microbes. Now dough, which is used for making foods such as dosa, idli, is also fermented by bacteria. Dosa, idli by bacteria. Okay, dough. The pulp of appearance of dough is due to the production of CO2 gas. Remember idli, dosa, CO2 gas. Now, can you tell which pathway is taking place resulting in the formation of CO2? Where do you think the bacteria for these fermentations come from? Similarly, the dough which is used for making bread is fermented using baker's yeast. So, dough is fermented using baker's yeast, that is the Saccharomyces cerevisiae. A number of traditional drinks and foods also made by fermentation by the microbes. So, toddy, a traditional drink of some parts of southern India, not northern India. Southern India is made by fermenting sap from palms. So these are the trees. Microbes are also used to ferment fish, soybean, bamboo shoots to make foods. Cheese is one of the oldest food items in which microbes were used. So different varieties of cheese are known by their characteristic texture, flavor, and taste. The specificity coming from the microbes that we use. For example, the large holes in Swiss cheese are due to the production of large amount of CO2 by a bacteria named Propionibacterium shermani. Okay, the Roquefort cheese are ripened by the growing a specific fungi on them which gives them a particular flavor. So this is the Roquefort cheese. Now Microbes in industrial products. So even in industry, microbes are used to synthesize a number of products valuable to human beings. So even in industry, microbes are used to synthesize a number of products valuable to human beings. Okay, beverages and antibiotics are some examples. Okay, in industrial products, production on an industrial scale requires growing microbes in very large vessels called fermenters, right? Now fermented beverages. Now you see, now the production of beverages like wine, beer, whiskey, brandy, rum. For this purpose, the same yeast Saccharomyces cerevisiae used for bread making and commonly called brewer's yeast, used for baking and commonly called brewer's yeast, is used for fermenting malted cereals and fruit juices. Fermenting malted cereals and fruit juices to produce ethanol. Now, do you recollect the metabolic reactions which result in the production of ethanol by yeast? Depending on the type of the raw material used for fermentation and the type of processing. So depending on the type of raw material for fermentation and the type of processing, it means with or without distillation, different types of alcohol drinks are obtained. Wine and beer produced without the distillation, whiskey, brandy, rum with the distillation. Okay, wine and beer without the distillation, right? So these are the fermenters and these are the fermentation plant. Okay, now antibiotics produced by microbes are regarded as one of the most significant discoveries of the 20th century and have greatly contributed towards the welfare of the human history. Anti is a Greek word that means against, and bios means living. Together they mean against life in the context of disease causing organism, whereas with the reference to human beings they are pro life, not against, because antibiotics are chemical substances which are produced by some microbes and can kill or retard the growth of the other disease causing microbes. So now commonly used antibiotic, that is penicillium. Do you know that penicillium was the first antibiotic to be discovered and it was chance discovery? Alexander Fleming, while working on Staphylococcus bacteria, once observed a mold growing in one of his unwashed culture plates around which Staphylococcus could not grow. Now he found out that it was due to a chemical produced. So it was due to a chemical produced by the mold, and he named it penicillium. So he was working on a Staphylococcus bacteria that was not growing because the mold was growing, which is the mold was Penicillium notatum, hence it is given the penicillium antibodies. However, its full potential as an effective antibiotic was established much later by Ernest Chain and Howard Florey. This antibiotic was extensively used to treat American soldiers wounded in World War II. Fleming, Chain and then Florey were awarded the Nobel Prize in 1945 for this discovery. Now, after penicillium, other antibiotics were also purified from other microbes. So after penicillium, other antibiotics were also purified from other microbes. Can you name some other antibiotics and find out their sources? So antibiotics have greatly improved our capacity to treat deadly diseases such as plague, whooping cough, colic acid, diphtheria, that is gal gotu, leprosy, that is kushtarog, which is used to kill millions of all over the globe. Okay, so chemical enzymes and other bioactive molecules. So chemicals, what are the chemicals like? Chemicals like organic acids, alcohols and enzymes are produced by the microbes. Examples of acid producers are Aspergillus niger, a fungus of citric acid. Then Acetobacter acidi, a bacterium of acetic acid. Clostridium butyrica, a bacterium of butyric acid. Lactobacillus, a bacterium of lactic acid. Yeast Saccharomyces cerevisiae is used for commercial production of ethanol. Microbes are also used for production of enzymes, right? Lipases are used in detergent formulations and are useful in removing oily stains from the laundry. So lipases are used in detergent formulations and are useful in removing oily stains from the laundry. You must have noticed that the bottled fruit juices bought from the market are clearer as compared to those made at home. This is because the bottled juices are clarified by the use of pectinases and proteases. So bottle juices are clarified by the process of pectinases and proteases and lipases, this removing the oily stains from the laundry. Now the streptokinase produced by the bacterium, that is streptococcus, and Now the streptokinase produced by the bacterium streptococcus and modified by genetic engineering is used as a clot buster for removing clots from the blood vessels of patients who have undergone myocardial infarction leading to heart attack. Another bioactive molecule, that is cyclosporin A, that is used as an immunosuppressive agent in organ transplant patients, is produced by the fungus Trichoderma polysporum. Another bioactive molecule, that is cyclosporin A, that is used as an immunosuppressive agent in organ transplant patients, is produced by the fungus Trichoderma polysporum. Another bioactive molecule, that is cyclosporin A, that is used as an immunosuppressive agent in organ transplant patients, is produced by the fungus Trichoderma polysporum. Statins produced by the yeast Monoscus purpurus have been commercialized as blood cholesterol lowering agents. So statins produced by the yeast Monoscus purpurus have been commercialized as blood cholesterol lowering agents. It acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol. So these statins acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol, right? So microbes in sewage treatment, right? So cyclosporin A, that is Trichoderma polysporum, right? Cyclosporin A, that is Trichoderma polysporum, and Monoscus purpurus, that is statins. So microbes in sewage treatment. We know that large quantities of wastewater are generated every day in cities and towns. Now these now 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 okay okay now so major component of this wastewater is human excreta. This municipal wastewater is called the sewage. Now it contains large amounts of organic matter and microbes. It contains large amount of organic matter, that is human excreta largely, and microbes. So many of which are pathogenic. So it is pathogenic. However, wonder where this huge quantity of sewage or urban wastewater is disposed of daily. This cannot be discharged into natural water bodies like rivers and streams directly. You can understand why. Before disposal, hence sewage is treated in sewage treatment plants, that is STPs, to make it less polluting. Treatment of wastewater is done by heterotrophic microbes naturally present in the sewage. Heterotrophic microbes, not autotrophic. Heterotrophic microbes naturally present in sewage. This treatment is carried out in two stages. So first stage is the primary treatment, second stage is the secondary treatment or biological treatment. So first stage, that is primary treatment. So this treatment stage basically involve physical removal of particles, that is large and small, from the sewage through filtration and sedimentation. So these are removed in stages. Initially, floating debris is removed by sequential filtration. Then the grit, soil and small pebbles are removed by the sedimentation. So in the first, large and small pebbles particles are removed from through filtration and this sedimentation, and the third, and all that and the remaining, that all solids that settle from the primary sludge, and the supernatant forms is the effluent. Okay, the effluent from the primary settling tank is taken for the secondary treatment. Now the secondary treatment or biological treatment. Okay, the primary effluent, now the primary effluent is passed into the large aeration tanks, where it is constantly agitated mechanically and air is pumped into it. This allows vigorous growth of useful aerobic microbes into flocs. That is masses of bacteria associated with fungal filaments to form the mesh like structure. That is the masses of bacteria associated with fungal filaments. The masses of bacteria associated with fungal filaments to form mesh like structure. These are the flocs. So these aerobic microbes, during they gets agitated and they form the flocs. Now while growing, these microbes consume the major part of the organic matter in the effluent. This significantly reduces the BOD, that is biochemical oxygen demand. Biochemical oxygen demand, not biological oxygen demand, is biochemical oxygen demand of the fluent. Now this reduces BOD. The BOD refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of water were oxidized by bacteria. The sewage water is treated till the BOD is reduced, not to zero. It is reduced till the BOD is reduced. The BOD test measures the rate of uptake of oxygen by microorganisms in a sample of water, and thus indirectly BOD is a measure of the organic matter present in the water. So thus indirectly BOD, biological oxygen demand, is a measure of the organic matter present in the water. Is a measure of the organic matter present in the water. The greater the BOD of wastewater, more is its polluting potential. So greater the BOD of the wastewater, more is its polluting potential, right? Once the BOD of sewage water or wastewater is reduced significantly, The effluent is then passed into a settling tank where the bacteria flocks. Now the bacteria flocks which are formed during the secondary treatment, biological treatment, now it's time to get sediment. These bacteria flocks are allowed to sediment. This sedimentation makes the activated sludge. Now small part of the activated sludge is pumped back into the aeration tank, which is the secondary treatment, that is biological treatment, to serve as the inoculum, to as a starter, to get enhanced, to fasten the secondary treatment, right? The remaining major part of the sludge, now the remaining major part of the sludge is pumped back into the large tanks called anaerobic sludge digester, where it gets digested. Here other kinds of bacteria, so flocks already have the bacteria and fungi, but now in large now in the anaerobic sludge digester, the other kinds of bacteria which grow anaerobically digest the bacteria and the fungi in the sludge, okay, which are the flocks. So during the now during this digestion, so now during this digestion so now during this digestion so here kinds of bacteria which grow anaerobically and digest the bacteria and the fungi in the sludge. So during this digestion, bacteria produces a mixture of gases such as methane, hydrogen sulfide and carbon dioxide. Methane, hydrogen sulfide and carbon dioxide. Now these gases form biogas and can be used as source of energy as it is inflammable. Now the influent from the secondary treatment plant is generally released into the natural bodies like rivers and streams. Okay, so you can appreciate how microbes play a major role in treating millions of gallons of wastewater every day across the globe. This methodology has been practiced for more than a year now in almost all parts of the world. Till date, no man-made technology has been able to rival the microbial treatment sewage. You are aware that due to increasing urbanization, sewage is being produced in much larger quantities than ever before. However, the number of sewage treatment plants has not increased enough to treat such large quantities, so the untreated sewage is often discharged directly into the rivers, leading to their population and increase in waterborne diseases. Okay. Now the Ministry of now the Ministry of Environment and Forest has initiated Ganga Action Plan and Yamuna Action Plans to save these major rivers of our country from pollution. Under these plans, it is proposed to build a large number of sewage treatment plants, that is STPs, so that only treated sewage may be discharged in the rivers. Okay. So this is an overview of a sewage plant, right? Now the microbes in production of biogas. So biogas is a mixture of gases that containing predominantly methane produced by the microbial activity and which may be used as fuel. So biogas is a mixture of gases. Biogas is a mixture of gases. Biogas is a mixture of gases produced by the microbial activity and which may be used as a fuel. So you have learned that microbes produce different types of gases and end products during growth and metabolism. The type of gas produced depends upon the microbes and the organic substrates they utilize. So the type of what depends upon the microbes they use. Type of gas produced depends upon the microbes and the organic substance they utilize. In the example cited in the relation to fermentation of dough, cheese making and production of beverages, the main gas produced was CO2. However, the type of organisms and organic substance they utilize, that depends. In cheese and in beverages, CO2 is produced, right? Now, however, certain bacteria which grow anaerobically, and in I think in idli and dosa there is puffed appearance, that is CO2 is produced, so it's okay, like, and in cheese also CO2 is produced, but I think it's SO2, right? Okay, no, it is the large holes in Swiss cheese is also the production of CO2 by the bacterium Propinium shermani, and okay, dup, idli, dosa fermented by bacteria, CO2 is produced. Okay, remember that thing. In bread making, it has nothing mentioned CO2 like that. Okay, so we are here. So however, certain bacteria which grow anaerobically on cellulosic material produce large amount of methane along with CO2 and H2. So produce large amount of methane along with CO2 and H2. So these bacteria are collectively called the methanogens because it anaerobically digests on cellulosic material. It produces CO2 and H2, and one such common bacterium is methanobacterium. These bacteria are commonly found in the anaerobic sludge during sewage treatment. So these bacteria are commonly found in the anaerobic sludge during sewage treatment, as we already know. Okay. These bacteria are also present in the rumen, a part of stomach of cattle. Okay, a lot of cellulosic material present in the food of cattle is also present in the rumen. Now in rumen, these bacteria help in breakdown of cellulose and play an important role in the nutrition of the cattle. Do you think human beings are able to digest the cellulose present in our foods? Thus, the excreta, that is dung of cattle, commonly called gobar, Thus, the excreta dung of cattle, commonly called gobar, is rich in these bacteria. Dung can be used for generation of biogas, commonly called the gobar gas. The biogas plant consists of a concrete that is 10 to 15 feet deep, in which the bio waste are collected and slurry of the dung is fed. A floating cover that is placed over the slurry, which keeps on rising as the gas is produced in the tank due to the microbial activity. The biogas plant has an outlet which is connected to the pipe, and all that. The spent slurry is removed through the another outlet. The cattle dung is available in large quantities. It's in rural areas, right? In rural areas, this is not in so biogas plants are more often built in rural areas. The biogas thus produced is used for cooking and lighting. It is used for the cooking, to make the food, is cooking, fire and lightning. Okay, electricity, lightning. The picture of a biogas plant is here. The technology of biogas production was developed in India mainly due to the efforts of the Indian Agriculture Research Institute, that is IARI, and Khadi and Village Industry Commission, that is KVIC. Khadi and Village Industrial Communities, right? Khadi and Village Industries Combination, the KVIC, right? So if your school, so this is, so microbe says biocontrol agents, right? So where we at, like, okay, so the typical biogas plant, as you can see, the technology of biogas production was developed in India mainly due to efforts of the Indian Agriculture Research Institute, IARI, and Khadi and Village Industry Communication, that is KVIC. Okay, now this is a typical biogas plant. You can see dung and water in here, and there is a digester, and then there is a gas holder which contains CH4 and CO2, and then there is a sludge, and then there is a gas and everything, this structure, right? Microbes as biocontrol agents. Now, biocontrol refers to the use of biological methods for controlling plant diseases and pests. In modern society, these problems have been tackled increasingly by the use of chemicals, by use of insecticides and pesticides. Okay, now these chemicals are toxic and extremely harmful to human beings and animals alike, and have been polluting our environment, soil, groundwater, fruits, vegetables, crop plants. Our soil is also polluted through our use of weedicides to remove weeds. Now biological control of pests and diseases. In agriculture, there is a method of controlling pests that relies on natural predation rather than introduced chemicals. In agriculture, there is a method of controlling pests that relies on natural predation rather than introduced chemicals. Okay, relies on natural predation. Okay, a key belief of the organic farming is that biodiversity furthers health. The more variety a landscape has, more sustainable. The organic farmer therefore was to create a system where the insects that are sometimes called pests are not eradicated, but instead are kept at manageable levels by a complex system of checks and balances within a living and vibrant ecosystem. Contrary to the conventional farming practices, which often use chemical methods to kill both useful and harmful. So in conventional farming, you can see we kill both useful and harmful life forms indiscriminately. This is a holistic approach that seeks to develop an understanding of the webs of the interaction between the myriads of organisms that constitute the field fauna and flora. The organic the organic farmer holds the view that the eradication of the creatures that are often described as pests is not only possible but also undesirable, for without them the beneficial predatory and parasitic insects which depend upon them as a food or host would not be able to survive, because the other insects that we need, if we kill in which these insects are dependent for food, That means we are killing both. If you are understanding this line is very deep. For without them, it is called the beneficial predatory and parasitic insects which depend upon them as food or host would not be able to survive. Thus the use of the biocontrol measures will greatly reduce our dependence on toxic chemicals and pesticides. Thus the use of the biocontrol measures will greatly reduce our dependence on toxic chemicals and pesticides. An important part of the biological farming approach is to become familiar with the various life forms that inhabit the field, predators as well as pests, and also their life cycles. So what we need to study the inhibitor, the field, predators as well as pests, and also their life cycles and pattern of feeding and the habitats that they prefer. This will help developing appropriate means of biocontrol. Now the very familiar beetle with red and black markings, the ladybird and dragonflies, are useful to get rid of aphids and mosquitoes. So red and black markings, ladybird is useful to get rid of aphids, and dragonflies is useful to get rid of mosquitoes respectively. An example of microbial biocontrol agents that can be introduced in order to control butterfly caterpillars is the bacteria Bacillus thuringiensis. To control the butterfly caterpillars is the bacteria Bacillus thuringiensis. Now these are available in sachets as dried spores which are mixed with water and sprayed onto vulnerable plants such as brassicas and fruit trees, which where these are eaten by the insect's larva. In the gut of the larva, the toxic is released and the larva get killed. The bacterial disease will kill the caterpillars, but but leave other insects unharmed. Because of the development of methods of genetic engineering in the last decade, so the scientists have introduced Bacillus thuringiensis toxic genes into plants. Such plants are resistant to attack by insect pests. These are the bt cotton is an example that are insect pest with the Bacillus thuringiensis toxin genes into them. Okay, so this is it. Now the now a bio now a biological control being developed for use in the treatment of plant diseases is the fungus trichoderma. Now trichoderma species are a, bi- a biological control being developed for use in the treatment of plant disease is the fungus. A biological control being developed for use in the treatment of plant disease is the fungus trichoderma. Now trichoderma species are free living fungi that are very common in the root ecosystems. They are effective biocontrol agents of several plant pathogens. Baculoviruses are the pathogens that attack insects and other arthropods. Now these are the baculoviruses that are pathogens and that attack insects and other arthropods, right? The majority of baculoviruses used as biological control agents are in the genus that is nucleopolyhedrovirus. The majority of the baculoviruses used as biological control agents are in the genus nucleopolyhedroviruses. The majority of baculoviruses used as biological control agents are in the genus nucleopolyhedroviruses. These viruses are excellent candidates for the species-specific narrow-spectrum insecticidal applications. So these viruses are excellent candidates for species-specific narrow-spectrum insecticidal application. They have been shown to have no negative impacts on plants, mammals, birds, fishes, or even on non-target insects. This is specifically desirable when beneficial insects are being conserved to aid in an overall integrated pest management program, or when an ecological sensitive area is being treated. Now this is IPM, that is integrated pest management, is being used when the desirable when beneficially insects are being conserved. We need to conserve the beneficially insects to aid in an overall integrated pest management IPM program, or when an ecological sensitive area is being treated. It means like we don't want to harm the other insects for killing like someone. We don't want to harm the beneficial insects that is used in the future for us. Okay, so this is how the baculoviruses are used. These are the species specific and narrow spectrum insecticidal application. They do not harm the others, they are species specific. So microbes as biofertilizers. Now, without our present day lives, the environmental pollution is a major cause, right? The use of the chemical fertilizers to meet the ever increasing demand of agriculture produce has contributed significantly to this pollution. Of course, we have now realized that there are problems associated with the overuse of the chemical fertilizers, and there is a large pressure to switch to organic farming, the use of biofertilizers. Now biofertilizers are organisms that enrich the nutrient quality of the soil. Okay, it increases the nutrient quality of the soil. The main sources of biofertilizers are bacteria, fungi, and cyanobacteria. Okay, now the nodules on the roots of the leguminous plants formed by the symbiotic association of ribosome. Think about the nodules on the roots of the leguminous plants formed by the symbiotic association of the rhizobium. These bacteria fix atmospheric nitrogen into organic forms, which is used by the plant as nutrients. Okay, other bacteria can fix atmospheric nitrogen while free living in the soil. So they can fix the atmospheric nitrogen while free living in the soil. Example, azospirillum and azotobacter, thus increasing the nitrogen content of the soil. Fungi are also known to form symbiotic association with plants, that is mycorrhiza. Fungi form the symbiotic association with plants, that is mycorrhiza. Many members of the genus, that is Glumomus, form mycorrhiza. So many members of the genus Glumomus form mycorrhiza with the fungi. I mean, the fungi forms with the association, symbiotic association with the plants. So fungi of the genus Glumomus, of many members, they form the association, symbiotic association with the plants, that is the mycorrhiza. Okay, the fungal symbiont in this association absorbs phosphorus from the soil and passes it to the plant. The fungal symbiont now the fungal symbiont now the fungal symbiont now the fungal symbiont in this association absorbs phosphorus from the soil and passes it to the plant. Now plants having such association show other benefits also, such as resistance to root-borne pathogens, tolerance to salinity and drought, and 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 overall increase in plant growth and development. So the plants having such association show other benefits also. The plants having such association show other benefits also, such as resistance to root-borne pathogens due to the fungi is providing all these, tolerance to salinity and drought, and an overall increase in plant growth and development. Can you tell what advantage the fungus derives from this association? They give food and shelter, maybe. Now, cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environments, many of which can fix atmospheric nitrogen. The cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environments. Cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environments. So, cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environments, many of which can fix atmospheric nitrogen. Example, Anabina nostoc and oscillatoria. Anabina nostoc and oscillatoria. In paddy fields, cyanobacteria serve as an important biofertilizer. So in paddy fields, the cyanobacteria serve as an important biofertilizers. In paddy fields, the cyanobacteria serve as an important biofertilizers. Right? Blue-green algae also add organic matter to the soil and increase its fertility. Now currently in our country, a number of biofertilizers are available commercially in the market, and farmers use these regularly in the fields to replenish all the and reduce the and the main purpose of it is to reduce the dependence on chemical fertilizers. That is the conventional farming that we see. So this is all your chapter summary is there.